Okay, we're going to swing into a new type of cell division, but this type of cell division is restricted in eukaryotes to just those tissues that are involved in sexual reproduction, in producing the sex cells, the ovaries and the testes. And so, what we're planning to do is describe the life cycle and chromosomes of eukaryotes, describe the process of making sex cells, which occurs during a specialized cell division called meiosis. And we'll talk about how meiosis produces genetic variability and why eukaryotes have sex. So whenever there's sex, I have some sort of picture in there to kind of represent that. So there I have James Bond from Russia with Love. So, a nice looking woman, nice well dressed man, two women fighting over a man, two men fighting over the woman, I guess. And then below that, I have the actual business. There's a human egg cell with sperm cell, and what you see behind it are the cells of the duct. That would be the oviduct where fertilization occurs. So to begin with, let's talk about, whoops, did I get that right? The eukaryotes, mitosis, and the cell cycle. So in the life cycle of eukaryotes, there's this long interphase period where cells appear to have a nucleus that looks the same throughout, and this is called interphase, but we know different things are happening in different parts of interphase. During the S phase, DNA is being copied, and in the G2 phase, the cell is getting ready for mitosis, getting ready to divide the nucleus and chromosomes, so it's producing extra chromosomes. In mitosis, the duplicated chromosomes are aligned in the center of the cell, and then they're pulled apart so that one sister chromatid goes each way. And we know that this is important because this is how cells or how organisms grow. They increase the number of cells. This is also used for the maintenance and repair of cells and healing after injuries. And if it goes wrong or it doesn't occur enough, you can get cancer or degenerative diseases. Then there are these checkpoints to make sure it occurs correctly. And these happen at the transitions from the G1 to the S phase. They allow the cell to assess the environment, to make sure the chromosomes have been duplicated correctly. There's one that happens before mitosis starts. And then there's a check right on the spindle fiber. Okay, and so this is normal cell division. This happens in almost all of your body cells. Okay, however, there's a specialized process for those cells involved in sexual reproduction. And for this, I should talk about eukaryotes and their genome a bit, about how many chromosomes they have and what exactly are the chromosomes. Eukaryotes are what is referred to as diploid. That means they have two copies of every chromosome. One they got from their father, one they got from their mother. And they will have two genes for each trait. And so this is why they're referred to as diploid, 2N. What's unusual is that in eukaryotes, right before reproduction, the organism makes gamete, where the chromosome numbers are reduced. And so sperm have half the number of chromosomes, and eggs have half the number of chromosomes. These two gametes combine to produce a new organism, a zygote, which will then have the diploid number of chromosomes. Okay, and so in eukaryotes, what we have are kind of an alteration of the life cycle between a haploid stage, which in humans and animals is very short-lived, it's just the sperm and the egg, and a diploid stage. So for us, the diploid stage is dominant and the haploid stage doesn't occur much. Okay, but in other organisms, it can be different. In some organisms, the main organism is the haploid form. The diploid form is temporary and goes through meiosis and only lasts for a short period of time. And so you see this in certain organisms. In other organisms, both the diploid and haploid forms are relatively long-lived, and you can find them associated with one another. They usually have a different appearance, but they're both present. So this seems kind of odd. Why have this kind of alteration of life cycles? Why have this? And this has to do with sex. This is sexual reproduction. Not bacteria don't do this at all. Neither do archaea. None of the prokaryotes have sex. And some of the eukaryotes do without it. They reproduce asexually, like many plants will reproduce by rhizomes or stolons, just spreading roots. They'll produce clones. Some animals will produce clones. And if you have a good genome, it makes kind of a sense. Like, why would I mix my genes with someone else's genes to produce offspring? It's kind of risky. It might break up good combination of genes. And so, like here we have a couple, and she's looking at him and thinking, are his genes worth this? Is it worth doing this? And the answer is we think so. That there are several ideas to explain why eukaryotes have sex. One is that by having 2 copies of every gene, if one isn't so good, you're probably still okay, and you may even be able to repair that bad one using the good one. Another example is it's just a genetic trick that's spread, so it's like a contagion. But a third answer, and probably the best answer, is that using this meiosis and this process of combining 2 genetic material from 2 organisms, you get a lot of genetic variability. And meiosis produces an astounding amount of genetic variability, which might be very good. If one of your offspring is susceptible to a parasite or a disease, it might be the others have more resistance. And so we think this is why eukaryotes have sex. And so meiosis occurs in the reproductive organs, in the testes in males, in the ovaries in females. Okay, in males we're making sperm, in females we're making oocytes. And an important thing here is to reduce the number of chromosomes. So what happens is you have 2 cell divisions with no DNA duplications between them. And you also have several mechanisms to get new combinations of genes. This is called genetic recombination. And so first, let me explain a little bit about chromosomes. Chromosomes, we have 46 chromosomes, but we're diploid. So what we really have are 23 pairs. And so one of each set comes from the father, one of each set comes from the mother. And we can see these in a karyotype, which is shown here. It's just a picture of the chromosomes that have been stained and kind of separated out so we can arrange them by size and staining properties and things like that. And so we have these 23 pairs, really 22 pairs, because the sex chromosomes are kind of different. The X is much larger than the Y. You can see it kind of here on the bottom. So these pairs are referred to as a homologous chromosomes, and each one is a homolog. It carries genes for the same traits as the one that came from the opposite parent, but it may not have the same genes. Well, in haploid cells, we get down to 23 chromosomes again. Okay, and how we do this is through meiosis. And in meiosis, these homologous pairs get duplicated, and then they associate together, and this is these are referred to sometimes as a tetrad. The process of them getting together is going to be really important. And then what's going to happen in meiosis 1 is one of these homologs will go one way and one will go the other. And in meiosis 2, the sister chromatids will separate. So here this cell had 2 chromosomes. Here we have 2 duplicated chromosomes. Here we have 1 duplicated chromosome. And here we have 1 unduplicated chromosome in each one of these final cells. That is the result. So something special happens when these duplicated chromosomes pair up. And this is in meiosis 1. This is called synapsis, where parts of the chromosomes overlie one another, and they exchange segments so that the chromosome becomes kind of a composite. Part of it came from the father, part of it came from the mother. And no longer are the 2 sister chromatids identical because they've exchanged material with their opposite. And so these things are going to separate, these homologous pairs are going to separate in meiosis 1, and then in meiosis 2, these duplicated chromosomes, the sister chromatids, will separate. So let's look at this in kind of a more detailed way. So here we see meiosis 1. Okay, before meiosis 1, the cell looks normal. It's an interphase. It has a nucleus with chromatin in it. The chromosomes are duplicated. In meiosis 1, the first thing that happens that's unusual is that homologous chromosomes pair up. And so instead of seeing individual X shapes move around, you see two X's moving together. They pair up and they exchange segments in synapses. And then in metaphase 1, these homologous pairs line up on the metaphase plate, and one of each homologous pair is separated. This is the reduction division, and so now the resulting two cells, instead of having two pairs of chromosomes, will only have one pair. Okay. Then in the division following that, meiosis 2, the sister chromatids separate. So here we have 2 duplicated chromosomes instead of 4, and these things will line up just like in mitosis, and sister chromatids will separate. But these sister chromatids are no longer identical. They are haploid, but they were all haploid right at the beginning of meiosis 2. So that haploid still happened in meiosis 1. So to summarize, all eukaryotes have 2 copies of each chromosomes. Humans have 23 pair of chromosomes. The sex cells in humans, sperm and egg, have just 23 chromosomes and are haploid. Meiosis produces the sex cells in the ovary, and meiosis has 2 rounds of cell division with no copying of DNA between. In meiosis, homologous pairs, chromosomes pair up and exchange segments in prophase 1. These homologous pairs separate in meiosis 1, but sister chromatids do not separate. All the cells are now haploid. All sex cells produced in meiosis are genetically unique because of the exchange of segments and the random shuffling. So here we have a picture here, 4 chromosomes in the original cell. Here's 4 duplicated chromosomes, 2 homologous pairs. The homologous pairs separate. And so here at the beginning of meiosis 2, we have just 2 chromosomes in the cell, but they're duplicated. And here sister chromatids separate. And so we wind up with 2 chromosomes in each gamete versus 4 in the original cell. Okay, we'll talk more about this. We'll talk about how it can go wrong in the next program.